Good morning. So I wanted to wake you up properly and things like that. That's why we have a bunch of statisticians to get you revved up for the rest of the day. And as Martin mentioned, I'm in bioinformatics at MD Anderson. I've been looking at high-throughput data for about 20 years. And I'm going to tell you some stories about that. And in particular, I'm going to talk about why I think reproducible research is really vital in the context of high-throughput biology. And it really boils down to a very simple observation, which is that when human beings start dealing with not just one variable or two or three, but with hundreds, our intuition about what makes sense in terms of how hundreds of variables behave, frankly, sucks. So we've got to do something to address that. Sorry, I'm going to briefly pause for an observation. First off, as an educational session, this is being recorded. This will be available after the entire conference. Second, all of my slides are posted on SpeakerDeck. You can download them. They're public. There is nothing secret about what I am going to present today. So you can quote, you can do whatever. So all of those are background, but the links are up there. The challenge, however, is that if our intuition isn't really that good, we're faced with a problem, which is that one of the reasons I got into working at the time with microarray data and other things like that was that we're hoping to say, can we find patterns of expression of groups of genes that will let us predict things like patient response or patient outcome, et cetera. But if I can't trust the intuition, as in does this set of 50 genes make sense, that means that in order to feel confident in using such a signature for clinical care, I have to be able to track or trace the individual steps that were used to assemble that signature so that because those are the things I can check. I know that this is a challenge empirically when I go to the literature, because actually one of the things that we've tried to do is every once in a while we'll read a paper in Nature or Science or Cell and we'll get really, really excited and we'll say, we want to implement that. We want to take it and use it to treat our patients better. And then we'll say, oh, well, okay, before we actually try rolling it out in a trial here, we should be able to just check the numbers they report because, hey, they've posted their data, they've posted the methods. It should be easy to check that and make sure that we can get everything they got, right? Has anybody here tried this experiment with papers from the literature, just basically taking it and saying, just given what they've told you, can you exactly reproduce the numbers? It's a non-trivial task. And actually, the problem that we've encountered is that every once in a while, even if it's hard to do, we've been backed into a corner because we really want to use it. So that has actually led us to develop a different subfield of bioinformatics, which we call forensic bioinformatics. And what this means to me is that, okay, here's the data they started with, here's the results they're reporting. Can we figure out what the hell they must have done to get from here to here, regardless of what the methods say they did? And this is occasionally informative, often though in ways we might not expect. And I want to take you through a worked example of this in the context of a clinical question of real importance. And that is particularly, the question I'm going to look at is, can we, for cancer patients, predict which drugs a given patient will respond to? How do I know this is important? If a woman shows up at MD Anderson Cancer Center today with ovarian cancer, we will give her standard frontline therapy, which is a platinum agent and a taxane. This will help roughly 80% of the women we give it to, in that it will shrink their tumor and they will feel better and their quality of life will improve. The other 20% we will poison for 6 weeks to no good effect. We will do this because we don't know going in what 20% they are. If we did, if we knew which ones wouldn't respond, we'd try something else. And that's the backdrop against which a paper appeared several years ago in Nature Medicine that got a whole bunch of attention because the authors, coming from Duke University, claimed to have largely solved this problem. So how did they do it? What did they do? They said, well, we're going to start not with patient samples, but actually with cell lines, and actually with a particular panel of cell lines that's been maintained by the US National Cancer Institute, the NCI, for about 40 years. And this panel of cell lines is known as the NCI 60. Now, why has the Cancer Institute gone to the trouble of maintaining this panel for 40 years? Because one of the ways that we figure out how these chemotherapeutic agents work is every time we have a new one introduced, the NCI takes that drug and they assay it for effectiveness against this panel of cell lines. They say, what concentration of the drug is required to cut the growth rate of this cell line in half? And this information about the cell line responsiveness to a given drug, this is public information. You can download it from the web. And this is available for every chemotherapeutic now in use. So what this means is that if we could in some way find a way to generalize from how cell lines respond to how patients respond, we have here the makings of a Rosetta Stone that would let us choose from all possible drugs. And that's what they did. Specifically, they said, well, we've got the sensitivity information. That's one piece. What's the other piece? The other piece is that we also have transcriptomic information for these same cell lines, again, public data, using the brand new tools known as microarrays, and in particular, Affymetrix gene expression arrays. And here's how it worked. They said, let's take a drug of interest. Let's say it's docetaxel. We'll go to the panel of cell lines. We'll take the most sensitive and most resistant, let's say 7 at each end, and then we will look at their microarray profiles, about 20,000 genes. Looking at all these things, gene by gene, we will apply immensely sophisticated mathematics, like 2-sample t-tests, to identify the most different genes. Then we'll take the top 50, and looking at the expression levels of the top 50, we will say the patterns, how they change going from sensitive to resistant. That pattern comprises the pattern of sensitivity to docetaxel. We will then look at a patient's microarray profile, look at just the values for those 50 genes and say, does it look more like a sensitive cell line? If so, give them docetaxel. More resistant, give them something else. Again, the cell line transcriptomic data is all public. The patient data that they used is all public. And when they tried this with 7 common chemotherapeutic agents, they reported getting great results every single time. And that meant that within weeks of this paper appearing, we had several different clinical groups at MD Anderson coming to us in bioinformatics saying, we want to do this. We want to use this method to treat our patients better. But the method involves math, and that's scary. So, can you help us? And we said, sure, we'd be delighted to help. We'd love to do it. We should be able to check what they did because it's all public. So we decided to try starting with a question we thought should be easy, which is, for a given drug, can we match the genes that they've found? The reason we thought that this should be easy is as a supplement to this paper. There's a series of tables saying for docetaxel, here's the genes that are important. For 5-fluorouracil, here's the genes that are important. And within the paper, they said, here's this gene, here's why it makes sense, and so on like that. So we thought it should be easy. So, okay, we picked 5-fluorouracil. They reported their top 45 genes. We tracked down the raw data. We did the 2-sample t-tests ourselves. I'm a PhD-level statistician. I can do this. And we got our top 45, and we said, the intersection, okay, what is it? Their top 45, our top 45, the intersection is 0. And we said, that's probably bad. What's going on? Well, it turns out that the genes, if you work with 20,000 genes, oftentimes we're not working with the individual gene names anymore. We're working with things like abstract identifiers. In this case, the abstract identifier is an Affymetrix probe set ID. It's somewhat numeric, but it's actually informative in this case because if what I'm showing you here in this plot are the probe set IDs of the first few genes they flagged as important, and here are the probe set IDs of the first few genes we find using 2-sample t-tests. Now, does anybody in the audience notice a pattern as we go from their list to ours? We have an off-by-1 indexing error. What happened? It turns out that we tracked down the software that they used. The software that they used requires 2 inputs. One is a matrix of gene expression values with 1 extra row for a header row saying, is the sample sensitive or resistant? And the second input is a list of gene names in the same order as the expression matrix, but for their software to work correctly. This list of gene names must not have a header row. But if you're working in Excel and you cut and you paste and you leave on this header row saying gene name at the very top, guess what happens? You get an off-by-1 indexing error. So what does this tell you? That they were able to find a story about why this list of genes made sense when none of the genes involved are actually due to the biology. This problem is present for the lists of genes that they found for every one of the 7 drugs. It got by the authors, it got by the reviewers, it got by the editors, because when we're talking about 50 or a few hundred dimensions, our intuition doesn't work. So we looked at this and we said, well, that's annoying. But while it's annoying and somewhat embarrassing, it's not fatal. It's not fatal because what we actually want to do is we want to use the numerical values to predict patient response. Adding on the gene names, that's something we do at the end. And they screwed that step up, fine. But if we can still use the numbers to predict patient response, we can still use this method. So can we use the numbers to predict patient response? So we said, let's set that as our goal. Let's try it. And to say, what is the goal for saying we can do it well enough? We said, if we can make predictions that are as good as the ones they got, then we're confident that we can use this. So that leads in turn to the question of how good were their predictions? So let me show you some of their predictions. And if we go to the top here, this is a panel from their paper where they're saying they're trying to predict breast cancer patients' response to single-agent docetaxel. And what they're looking at in this cohort is they say most of the patients who responded to treatment, those are the blue squares here, they had low scores, that's good. And most of the patients resistant to treatment, red triangles, they had high scores, that's good. There's pretty good separation. Now, they didn't get them perfect. There's these 2 blue squares that got high scores. But overall, the accuracy, they say, is we got 22 out of 24 right. Not perfect, but still pretty darn good. So we looked at that and we said, okay, that's great. Data's public. But I have a deep analysis question for the audience. How many blue squares are there in this plot? There are 13 blue squares and 11 red triangles. That's the 24 they got. 13 sensitive and 11 resistant. But I told you that all the data they used was public. So we went out and we tracked down the raw data set, which showed up in a paper in The Lancet a few years ago. And they spent a lot of time talking about their 11 sensitive and 13 resistant patients, not 13 and 11. This got us a bit concerned. So we decided to take a look at the results from another paper. And for those of you who are really ambitious, how many red triangles are there? Okay, maybe not. But basically, this is another one where they claimed great success, where most of the patient population, most of the patients were resistant to treatment. The problem, the source data for this, in this case, came from a paper dealing with childhood ALL. This is a form of cancer which actually we know pretty well how to treat. We can cure roughly 80% of the kids who get it. But what this means is that if in this patient population we have a drug to which most of the patients are resistant, we would not be using it. So why do they think most of their patients are resistant? Well, the actual authors of the raw data thought that most of their patients were sensitive. Well, if we go back and we look more closely at the software that they're using, I told you that it required a gene expression matrix with a label saying, is it sensitive or resistant? Well, actually, the labels that they used were 0 or 1, but they never explicitly wrote down 0 equals sensitive, 1 equals resistant. So in some cases, going from one drug to the next, they got the labels reversed. So what does this mean clinically? It means that if we were to use this method, we would wind up administering adriamycin just to the patients who are most resistant to it, which is poor clinical practice. We do not advocate this. We think this is a bad idea. Now, this stuff that I've just told you about, this actually took a bit more than a few minutes to figure out. This was actually the result of about 6 months of interactions between us and the original authors saying, we'd love to get this to work. We're trying to get this to work. Can you help us? We have some questions about the data. And after these 6 months, we basically said, well, we're really concerned because actually if we use the labels which we believe to be correct, the ones that actually track with the raw source data, we don't think your method works. At which point the initial authors said, well, obviously this interaction has ceased to be productive, so we wish to draw it to a close. And we said, okay. At which point we wrote up our various objections and sent it off as a letter to Nature Medicine, where the paper had initially appeared. And our letter appeared that November saying, here are the various problems. And at that point, we also got to see the author's response. And we were sort of wondering where they could say, oh, we're screwed up, we're so sorry, we'll never do it again. And that really wasn't the tone of the response. The response was more that, no, the analyses by Baggerly and Coombs are deeply flawed. Our analyses are correct. We've gotten our sample labels right, the one with all the red triangles, that's adriamycin. All of our labels are correct. We've posted new data to our website to show this. Furthermore, we have continued to apply this method in other high-profile papers with editorials and things like that, and we continue to get great results, so we must be right and they must be wrong. Okay, well, maybe we screwed up. They said they posted data showing that they were correct. So we said, all right, let's take a look at that adriamycin. That's the one with all the red triangles. They posted new data to their website. What did they actually post? They posted a big Excel table with 144 columns, and the columns had two headers. One header said, is this a cell line or is it a patient sample? The second sample said, is it resistant or is it sensitive to treatment? Okay, great. And we count the numbers of resistant patients, and it matches the numbers of red triangles. Okay, maybe that has something. Okay, well, the other thing, if we've got all the data, maybe we can take a look at clusters. So we checked the pairwise correlations between samples, and looking at all 144 samples that they post, here's a plot of where the correlations are really, really high. Down here, this square, these are the cell lines. This line of red dots along the diagonal, that says that cell lines are highly correlated with themselves. I believe that. That's good. That we want to have. But then we get to the patient samples. This main diagonal line we expected. These we didn't. What the heck's going on? Well, I told you that there were 122 columns of patient data. Well, there are 122 columns, yes, but the contents of those columns, the numbers, are not all distinct. There are some columns of numbers that they have reused 2, 3, or even 4 times. And right about here is one patient's data where they have used that patient's data 4 times, and they have labeled that patient resistant 3 times out of 4. So we're pretty confident they don't have the labels right yet. So we sent a note about this to Nature Medicine. Nature Medicine said, oh, well, the authors will make a correction. There's no need to publish your objection. So indeed, the next year, the authors made a correction where they said, we've looked at it using only the 95 unique samples, and again, we get great results. We were a bit surprised because when we'd counted, we'd only seen 84 unique samples, so 95 was a surprise to us. So when we look at the 95 that they're using, it turns out of the 95 that they're using, there are only 80 that are distinct. 15 are present twice. Of the 15 that are present twice, 6 are labeled both sensitive and resistant. So the correction of a correction still needs correcting. But Nature Medicine had said it was done, so we decided to move on. We decided to move on next because they said they'd gotten it to work in other papers to carry it forward. One of the other papers was in the Journal of Clinical Oncology, and it deals with 2 new drugs, cisplatin and pemetrexed, which are commonly used in the treatment of lung cancer. They said, we've gotten great results here, again, and something that persuades us that this makes sense is that when we look at the signature that we've obtained for cisplatin, some of the genes that are contained in this list are ERCC1, ERCC4, and DNA repair genes, and there have just been a few papers in the New England Journal saying that this is how cisplatin acts. These are the pathways it acts through, so this is persuasive to us. So the folks in the lung cancer group at MD Anderson said, could you check this? So we went through, and for cisplatin and pemetrexed, with some effort, we reverse engineered our mappings of the gene list. And for pemetrexed, we can match all 80 of the genes they report after we account for the off by 1 error, which is still there. For cisplatin, we can match 41 of the 45 genes they report after we account for the off by 1 error. There are 4 that we cannot match at all, that the numbers do not supply. Do I have any guesses from the audience as to the identities of the 4 probe sets we can't match? Turns out that the 4 probe sets we can't match map to ERCC1, ERCC4, another probe set for ERCC1, and another one for FANC-M, which is DNA repair. So these are the 4 that are most persuasive to them, but we can't get it from their numbers. Even better, even in this context, the last 2 probe sets I've listed here are special. What makes those 2 special? Well, it turns out that those 2 probe sets were not physically present on the arrays that they used. This was actually part of a two-array set, the U133A and the U133B. These 2 at the bottom are from the U133B, which was never run. So 2 of their best genes have never been measured yet. We thought this was a neat trick. We sent a note about this off to JCO. JCO said, we're sorry, we can't publish this. We sent a second note saying, could you tell us why? They sent us a second note saying, we're sorry, we can't publish this. So we decided to move on. We moved on instead to the other paper they cited as validation of their approach. This one showed up in the Lancet Oncology, where here what they were doing was they were extending the approach to deal not just with one drug, but rather with combination chemotherapy. This is important, of course, because most cancer patients, we hit the tumor many different ways. So what were they looking at here? They were looking at ER-negative breast cancer, and they were saying, we're going to compare 2 combinations. Combination 1 is FEC, fluorouracil, epirubicin, and cyclophosphamide. Combination 2 is TET, Taxotere followed by epirubicin and taxotere. It's just 2 drugs. The fun thing here is that they say if we had used their rules to allocate patients to treatment arms as opposed to randomizing them, which is what was done, we would have improved the response rate from 44% to 70%. That's a big jump. So we looked at it and we said, okay, great. We've got the data. It's all public. Let's take a look. Maybe if we cluster the data, we can see 2 big clusters that say, here's the responders to FEC, here's the responders to TET. So if we look at the data, do we see 2 clusters in the data? Well, here's the raw data. Here are the cases where the correlations are really high. Everybody sees 2 clusters, right? Maybe 2 plus 1 or something like that. So we looked at that and we said, what's going on? Well, actually, if you look at the raw data for Affymetrix and for a few other files, there's really informative stuff there. And the raw data here is known as cell files. These are mostly text files that have the expression measurements for every gene, but they also have some extra information at the top. That header information includes, amongst other things, what day was this assay run? So on the right, I've taken this same set of arrays and I've plotted them as a function of run date. So this first block right here corresponds to these samples, which were all run around the same time. The black circles, those are half of the patients treated with FEC. Green X's, those samples were excluded from the final analysis for one reason or another. This second block, that's the other half of the patients treated with FEC. This third block, that's all of the patients treated with TET. We have complete confounding of the experimental design. All of the patients treated with one drug combination, all of their samples were run months after all the others, as it happens in a different lab in a different country. Some of the differences we're seeing here, and there will be some, are not driven by biology. They're driven by differences in processing. That's a problem. So we said, okay, we're not too happy about the experimental design, but still they say they found something, maybe we can still exploit it. But the other thing that really bugged us, and we were sort of curious about, was how are they getting predictions for combinations? We sort of understood the mathematics for predicting one drug at a time. So we said, okay, they're predicting probability of response to one drug at a time. How are they dealing with combinations? Well, we went back to their Nature Medicine paper, and in their Nature Medicine paper, they said, in outline, here's how it might work for a different combination. And they listed a 4-drug combo, TFAC. So we actually have 3 different possibilities here. So we have a 4-drug combo, a 2-drug combo, and a 3-drug combo. So what formulas are they using? This is the only slide in the entire talk with formulas, so here you go. So for TFAC, here's the rule that they used. The probability that a patient will be sensitive to this 4-drug combo is given by the probability they will be sensitive to T plus P of F plus P of A plus P of C minus the product of all 4. Anybody in the audience see a problem here? The authors saw a problem because all of the probabilities they got using this rule were bigger than 1. So they said, well, we know the biggest one should be 1 and the smallest one should be 0, so we'll set those and we'll linearly interpolate everything else down from there. Now, this step, oddly enough, did not make it into the methods section of the paper. The way we know this is we tracked down the Excel file they were using, moused over the cell, and said, oh, that's the rule they're using. Okay, fine. Now, this is not really a great rule, but still, as they go on to the other rules, like TET, this is a 2-drug combo. Actually, this formula would be okay for 2 drugs. It's the independence rule for the combination of events. They've just generalized it incorrectly. So what is the rule they used for 2 drugs? It's different. Now, it still gives you numbers between 0 and 1, but that's one thing. Okay, what about the 3-drug combo? I will buy dinner for the first person who can explain to me where the hell this rule comes from. I have no clue. But what I can tell you is that every single rule is different. And to paraphrase a rather famous American, I have a dream. And my dream is this, that if you are going to attempt to validate a clinical prediction rule in new patient cohorts, that you would keep the prediction rule the same. So the fact that they didn't bothers me. Okay, so we're not happy with the design, we're not happy with the combination rules, maybe we can still get something. So in particular, maybe if we just looked at one drug at a time, we could still make something useful. And actually, they tried this. They said for individual drugs, if we try making the predictions just based on those, can we predict patient response? And they said for some of them, the answer is yes. So if they're trying to predict response to FEC treatment using just the results for single drug response to C, which is Cytoxan, here is the ROC curve that they report. And this is a beautiful ROC curve. And we said, wow, that's great. And then we tracked down the data and we fit our own ROC curve and we got this, which looked a bit different. And then we said, okay, well, that doesn't really make sense. Can we figure out what's going on? And then we actually asked a different question, which is, Cytoxan. I have a biological question. And are there any doctors in the audience here? Medical doctors? Anybody? Well, how about a different question? Anybody here know what a prodrug is? I hear laughs, so do you know what it is? Well, a prodrug is a drug that is not active in and of itself. It's processed by our bodies into the active form. And actually, the fun thing here is that Cytoxan is a prodrug. It's processed by our livers into the active form. So the follow-up question to that, of course, is what do you think happens if you give Cytoxan to a bunch of cell lines that don't have livers? Absolutely nothing. So how on earth did they identify the cell lines that were sensitive and resistant to Cytoxan? Well, we took a look at the cell lines that they were using more closely. It turns out that these are the cell lines that are sensitive and resistant to pemetrexed. They got the drug wrong, but they still got great results, so who cares? So we wrote this up as a series of objections. We sent this off to the Lancet Oncology, and Lancet Oncology said, well, you are obviously arguing about statistical issues to which everyone knows there are no right or wrong answers. So there is no need to publish your objection. So we said, okay, and we moved on. We moved on because some of the folks we were working with dealing with glioblastoma, brain cancer, said, well, they've now dealt with temozolomide, and they claim great results there. Here's the heat map that they get trying to predict patient response. Can you check this? So we took a look at the data they were using, and we said, can we match this heat map? And we tried for a good while, and we couldn't match it. We were getting very frustrated, so we said, maybe we just don't understand what's going on. So we went back to some of the earlier papers to say, can we figure out from the earlier papers what's going on? And in doing so, we looked really closely at the heat map they'd reported for cisplatin 2 years before. And we looked very closely at these two heat maps and said, you know what, we're not so bothered by our inability to match it with temozolomide anymore, because it's exactly the same, and that shouldn't happen. So we sent a note about this off to the editors of CCR and JCO and said, what's going on? And the editor of CCR wrote back and said, well, the authors submitted the wrong figure, no problem, we'll fix that, and no need to publish your objection. Well, the authors then supplied a new heat map and a new gene list, which did not agree with each other, by the way. And in this paper, they have now achieved a neat hat trick of it being nominally about temozolomide drug A, having a gene list for some other drug, and for a heat map for yet a third drug. 3 drugs, all not really showing what we want, all in one paper. Now, all of this took course over this course of several years trying to figure out what's going on because we wanted to use it. But why the hell do we care? Well, the reason we really care is that a few years after all this began, we learned that the investigators at Duke had already started using this rule to allocate patients to treatment arms in clinical trials they were running at Duke. In some of the clinical trials, the drugs that they were using were ones for which, based on the data they'd posted, they had the sensitive and resistant labels reversed. We were not happy. We wrote up our objections. The first thing we did with that, we submitted it to the editor of a prominent biological journal for informal comment. His response, your story is too negative. Can you fix that? And we said, you know what? We really don't think we can. So the next thing we did was we sent it to the Annals of Applied Statistics. That's okay, most doctors read that one too. And the response there was a bit different. We got email from the editor that evening saying, you know what, I like this paper, which we took as a good sign. And we got another email from him 2 weeks later saying, you know what, the AEs and I have just decided to accept the paper as is. We've posted it online now, which means that I'm sending more papers there. But the other thing here is that, okay, the paper's now in the literature, which means we can cite it, and means in particular we can talk with the press. So we went then and we talked with the editor of The Cancer Letter, which is a journal which is occasionally read by the cancer community. And a few weeks later, Paul Goldberg of The Cancer Letter ran a story saying, biostatisticians at MD Anderson are alleging the potential for patient harm in clinical trials being run at Duke University. Oh, the horror! And it was wonderful and things like that. And unbeknownst to us, the National Cancer Institute read our paper, and they looked at grant applications from the same group, and they said, we see the same mistakes here. So the NCI sent a side note to Duke's Institutional Review Board, that's the group in charge of ensuring patient safety, saying, we are concerned. Will you look at this and reassure us? Now, I'm a professor at a different institution. They may be annoyed by me, but they're not going to do too much. The NCI is funding them to the tunes of tens of millions per year. They'll pay attention. So Duke said, you know what, we're going to suspend enrollment in these trials. We're going to convene a scientific expert review panel. We're going to look at this, say, is the science justified going forward? And we breathed a sigh of relief saying, we're sorry, we're really upset that it got this far, but given that it did, this looks like a reasonable way to address it. So we said, okay, what's going to happen next? So while things were suspended, the investigators at Duke posted brand new data. What data did they post? They posted the quantifications for the samples they'd used to validate their signature for cisplatin and pemetrexed. These are the ones that proved it worked. The samples that they were reporting on were 59 samples from a larger public data set involving 153. We knew where it was. We said, great, let's look at it. So we downloaded all the samples, we checked this correlation, and we said, well, here's the 59 labels that they report, here's the 153 samples. If the samples are what they say they are, what you should be seeing in this plot are 59 red squares all falling on top of this blue line. How many red squares are on top of that blue line? 0. As it turns out in this plot, there aren't even 59 red squares. There's 43. That means that for those 43 samples, we can match the numbers exactly. We know what the source sample is, and it's not what they say it is. They got the sample label wrong, and as a result, they got the clinical annotation wrong. For the other 16 samples, there's no red squares. Why not? Because for those samples, they've already scrambled the gene name so badly that we have no clue what these samples are anymore, but we can tell you they're not valid. And more than that, all 59 validation samples are wrong for drugs they've already been using in clinical trials for 2 or 3 years. And we wrote a report about this, and we sent this to Duke and the NCI, and we said, what do you want to do? The next thing that happened was that Duke announced they were getting ready to restart the clinical trials because they had concluded their scientific investigation, and their investigations and results had strengthened their confidence in this evolving approach to personalized cancer treatment. And we were surprised, and we said, would you show us the data upon which this decision was based? And Duke said no. Duke said, while the scientific reviewers approved of our sharing the report with the NCI, we considered a confidential document. A future paper will explain our methods. We're going to treat the patients this way now, but we're going to explain why it works later. We're not too happy about that. But something Duke actually said gave us a clue, gave us a different avenue of attack, which is that while the reviewers approved of our sharing the report with the NCI, if we pause there, the National Cancer Institute is a federal agency, which means that in the United States, it is subject to the provisions of the Freedom of Information Act. We don't have to get the report from Duke. So indeed, a few months later, we were able to get a copy of the report from the NCI pursuant to a FOIA request. And we looked at the report and we said, this does not justify restarting trials. What the hell are you thinking? And Duke had no comment. And at this point, we were just going, is there anything else we can do? We're just getting sort of depressed here. And then, one evening, I was sitting in my office and I got a phone call. And I got a phone call from a guy who said, hi, I'm a lawyer. My client wishes you to be aware of the following information pertaining to some of the investigators at Duke. And I chatted with him for a while, and I said, you know, that's really interesting. Here's the number of a reporter you should call. And about an hour later, I got a call from Paul Goldberg of the Cancer Letter, and Paul said, I'm going to have fun. So why was Paul going to have fun? Well, a few weeks later, Paul wrote the story where he pointed out that one of the investigators at Duke, Anil Poti, used to claim on his CV and on various grant applications that he was a Rhodes Scholar, but the Rhodes Foundation disagreed. So what we know in particular that Poti said, well, maybe I should talk with this reporter. After all, I was a nominee. We know this because Paul had actually emailed him a set of questions, and he'd CC'd a bunch of deans in the Duke administration when he did so, and Poti hit reply to all. So what has just happened? You've just had a member of your faculty admit in writing to a member of the press that, oh yes, I've been committing fraud for the past 5 years, and CCing a bunch of the university's administration when he admits this fraud. He was placed on administrative leave that day. That was Friday. Now, I got in touch with a bunch of friends of mine, and I spammed them with this article. And I said, I know some of you have been listening to me about this for years. I know some of you have been talking about writing letters and things like this. Now is politically the best time you will ever have. So, the following Monday, a letter from a whole bunch of statisticians landed on the desk of Harold Varmus, the brand new head of the National Cancer Institute, saying, would you please resuspend the trials so that we can figure out what the science is? And Varmus called Duke and he said, you're going to suspend the trials? And Duke said yes. And Varmus called the Institute of Medicine and said, would you act as an impartial reviewer? And the IOM said, yes, we'll start in December. And things were set to go from there. But before we got to December, well, actually, Joe Nevins, the senior author on many of the papers, sent email to several of his authors saying, you know what, we may have to pull one of the papers. What paper were they going to have to pull? One dealing with cisplatin and pemetrexed. Why were they going to have to pull it? Because on closer examination of their database, it appeared that 43 of the validation samples were mislabeled and they could not find the other 16, which is what we told them the previous November before they enrolled more patients. Week after that, Duke officially terminated the clinical trials and announced that in retrospect, these trials should never have been run. A few weeks after that, Anil Poti resigned from Duke and a cascade of retractions and other things began. So other things, well, there were, in this particular case,
100 or so patients in these clinical trials. Shortly after this all broke down, the lawsuits began. The retractions were from such fly-by-night journals as the New England Journal, JAMA, and things like that. This hit the front page of the New York Times. This hit the television show 60 Minutes. And actually, I wasn't able to get the video working, but we're going to see if I can play the audio for you from just a brief clip from that one. So we'll see.
Dr. Nevins discovered that when the underlying data disproved Dr. Cody's theory, the data were changed.
It became clear that there was no explanation other than there was a manipulation of the data, a manipulation of somebody's credentials, and a manipulation of a lot of people's trust.
Manipulated data. These were not errors.
That's correct. It simply couldn't be random. It simply couldn't be inadvertent. It had to have been based on a desire to make something work.
Is it a close call, or is it abundantly clear that the data were fabricated?
Abundantly clear.
It is abundantly clear that the data were fabricated 5 years after clinical trials began. The data were not examined to find this abundantly clear falsification until after everything crashed down. That's an incorrect sequence. But more to the point, the case that I have just described for you, this is pathological, let there be no mistake. We do not see this many mistakes in very many papers. But we recognized the component mistakes. How are we able to do this? Because I've been working at MD Anderson for 20 years, and I will tell you that I have seen each and every one of the types of mistakes I just reported, mislabeling of samples, confounding in the design. I've seen them all in data sets that have come to me at MD Anderson. MD Anderson is a really, really good institution. It's just that these types of mistakes, simple mistakes, are easy ones to make. They're particularly easy mistakes to make if you're working in Excel and you're cutting and you're pasting, and every once in a while you leave out a row and the contents of the column shift up by 1. But that means that if they're simple mistakes to make, if you get all the documentation, and at MD Anderson we can ask for it, we can typically fix it really quickly. It's easy to do. But if you take that problem of simple mistakes and then you say the journal won't let me publish all the supporting documentation, and you decouple that incorrect result from the supporting information, then you have literature which has problems, and that's something we're worried about. Because the broader question is, I just told you about a particular set of papers. How broad spread is this? Does this affect the broader scientific literature? And there have been a few other survey papers that have been looking at this. Probably one of the ones that was most scary showed up in 2012 in Nature by Glenn Baggerly and Lee Ellis. Glenn Baggerly was the head of clinical development at Amgen. He said, working with the initial authors of 53 breakthrough papers, we tried to replicate the findings so that we could commercialize them. Working with the authors, we were able to replicate the results for 6 of the 53 studies, just a bit over 10%. This has the NIH and various other funding agencies really scared and upset. They don't want to go to Congress and say, yeah, by the way, most of the money we're spending is wasted. They don't like that. So in particular, as of 2016, the NIH said, we're going to start imposing rigor and reproducibility requirements on all of our grant recipients. Here's the types of money we're talking about. The NIH research budget is roughly $50 billion a year. They think half of that is wasted. How is it wasted? Biological reagents and reference materials. This is, did you get the labels right? Study design. Did you avoid confounding? Data analysis and reporting. Did you tell us what you did? 90% of $25 billion is lost due to simple mistakes we can avoid if we're aware of them. Now, we were asked what we might recommend. The NCI was asked what they would recommend. The NCI actually issued a set of guidelines in 2013 saying, here's the types of things we want for any trial we fund. This came out in Nature in 2013. They'd planned a press conference, but they couldn't talk about it because it happened when the paper came out. The government was on furlough, and none of the authors could talk or they would lose their jobs. But we were also asked what we want to see in a high-throughput paper, and quite frankly, at this point, we want to see the data and we want to see the code because we have now passed the point where verbal descriptions are adequate. We need to see what you did. But it's actually a bit blunter than that. Specifically, if you're publishing a paper in high-throughput biology, by the time you publish a paper, there's really a succinct way of expressing what I want. at the time of publication, you're saying, I don't have to trust you anymore. It's perfectly reasonable for me to ask. So I think I should. Now, believe it or not, I'm an optimist. And the reason I'm an optimist is that I've been doing this for 20 years, and I'm going to tell you the data is so much better than it was 20 years ago. And the reason that I'm really optimistic is that the tools for doing it right have gotten better and easier to use. More to the point, if you want to see, so I've just given you a short intro, and in a short intro, my basic sales job is to convince you that this is important. If I want to tell you how to do it right, I'm sorry, it takes me a bit longer to tell you how to do it right. So this is actually the general outline of a 4-hour short course. I said, here's the basics of how you do it right. This is a link to the recording of that short course, which should be live sometime this coming week. So if you want to learn more, here's things like that. Furthermore, here are some links to other places you can learn more about this. I do have to acknowledge that I didn't do all of this myself. First among equals is Kevin Coombs, who helped me with this. Between the two of us, doing all this reverse engineering took us a bit over 2,000 man-hours. That does not scale as a way to correct the literature. And in particular, it's more time than you want to spend on analyses that should have already been done. But the last thing I need to do is I need to thank all of you for putting up with me for 40 minutes. That's what I got. Thank you, folks.